Here we're going to discuss the process of pulmonary ventilation and movement of gases between the blood and the tissue. So the process of pulmonary ventilation is governed by so-called Boyle's law, which is exemplified in this picture right here, that when pressure changes, volume changes in opposite direction. When pressure, well, in our case, when volume changes, pressure changes. When volume increases, the pressure in that volume decreases. When volume decreases, the pressure in that volume increases. The process of the respiration is governed by intercostal and diaphragm muscles mostly. There are plenty of accessory muscles like sternocleidomastoid or scalenes, but we're not going to address those today. So when you inhale, diaphragm and intercostal muscles contract, and that expands the thoracic wall. When thoracic wall expands, lungs expand as well. When volume of the lungs goes up, the pressure in the lungs goes down and air rushes into your lungs. When you exhale, diaphragm and intercostal muscles relax. The volume of the lungs decreases. Pressure in the lungs, volume decreases, pressure increases, and that pushes the air out. So what are the pressures that drive the air in and out of the lungs? Atmospheric pressure doesn't really change. We can assume that it's always 760 millimeters of mercury. Now, alveolar pressure is the pressure in the alveoli, basically pressure inside the lungs. It is also called intrapulmonary. Alveolar pressure is the same as intrapulmonary. Now, intrapleural pressure, shown here, is the pressure inside of the pleural cavity. Alveolar or intrapulmonary pressure is always higher than intrapleural pressure. If alveolar pressure becomes equal to intrapleural pressure, your lungs will collapse because they are elastic, just like the balloon will collapse if the pressure inside the balloon will be equal to the pressure outside of the balloon. So when you inspire, the pressure in your lungs, your alveolar pressure, becomes lower than atmospheric pressure, and the air rushes into your lungs. When you expire, your alveolar or intrapulmonary pressure will become higher than atmospheric, and that will force the air out of your lungs. And as I mentioned before, if intrapleural pressure and intrapulmonary or alveolar pressure become equal, if these two pressures for some reason become equal, then your lung will collapse. The structures of the brain that regulate how fast you breathe are located in the pons and medulla oblongata. So essentially the central structure is so-called ventral respiratory group. It actually sets up the normal breathing rate. So it stimulates the contraction of the muscles, diaphragm and intercostals, and then relax, and then contracts and then relax. So that's how your eupnea or normal breathing rate is set up. Dorsal respiratory group and pontine respiratory group, they regulate your breathing rhythms when you, for instance, exercise or you talk or you sing. Chemical stimuli have a really important role in regulating our respiratory rate and the depth. Essentially, there are three chemicals that play the main role. It's carbon dioxide, pH or acidity, and oxygen in the blood. So chemoreceptors in the aorta and carotid artery sense the levels of these chemicals and send signals to the brainstem. In response, brainstem sends the output signals to the muscles, and the muscles increase rate of respiration and depth of respiration. So what is the most important factor that increases respiratory rate and depth? Most important factor is elevated levels of carbon dioxide in the blood. These elevated levels of carbon dioxide in the blood will stimulate more frequent and deeper breathing, and you will be able to exhale all that excessive carbon dioxide. Second, in terms of its efficiency, in terms of its influence on the respiratory rate and depth, is pH. Low pH acidosis significantly increases the respiratory depth and respiratory rate. Low oxygen isn't that important. It does increase respiratory rate and depth, but it's the third in terms of importance. Now, what are normal respiratory volumes? So there are several. We're going to discuss the volumes and we're going to discuss some lung capacities. So respiratory volumes, let's talk about these first. Here, this one is the tidal volume. That's the normal breathing. That's what you regularly breathe. Forced inhalation is the term that refers to the volume that you can inhale after the tidal volume. This is called inspiratory reserve volume. This is when you try to fill your lungs to their top. Now, when you exhale, and then you try to exhale even more, so the volume that you exhale after normal tidal exhalation is called expiratory reserve volume. There is always some air in the lung. That volume of air that always stays in your lungs is called residual volume. These four volumes form four lung capacities. Total lung capacity is the total volume of the lung, all four volumes pulled together. Vital capacity reflects the amount of air that you can manipulate, and it's called vital capacity for that exact reason. Inspiratory reserve volume, tidal volume, and expiratory reserve volume form vital capacity. The amount of air that you can inspire after normal tidal exhalation is called inspiratory capacity. It is formed by inspiratory reserve volume and the tidal volume. And functional residual capacity is the amount of air that's left in the lung after you exhale. Expiratory reserve volume plus residual volume. So this table represents these volumes all together. With that said, we're going to be moving to the gas exchange in the processes of external and internal respiration. The processes of external and internal respiration are governed by so-called Dalton's law. Dalton's law suggests that total pressure of a mixture of gases equals the pressures of each individual gas, so-called partial pressures of gases combined. And partial pressures are proportional to the gas concentration. So gases move independently, meaning that the diffusion of oxygen does not depend on the diffusion of carbon dioxide and vice versa. And they always move from higher pressure to the lower pressure. Now, in the external respiration, you can see the process happening here. Oxygen move into the blood. Why? Because alveolar pressure of oxygen is 104 and pressure of oxygen in the blood is 40. 104 is obviously more than 40, so oxygen diffuses from high pressure to the low pressure. So in the pulmonary capillaries, oxygen diffuses from alveoli into the blood. Now for carbon dioxide, the picture is opposite. The pressure of carbon dioxide in the blood is 45, and the pressure of carbon dioxide in the alveoli is 40. So carbon dioxide diffuses from the blood into the alveoli. Once the equilibrium is established, blood, highly oxygenated, moves towards systemic capillaries. And what happens in the systemic capillaries? Well, concentration of oxygen in the tissues is lower than 40. Concentration of oxygen in the blood that gets to the systemic capillaries is 100. So oxygen goes from the blood into the tissue cells. And for CO2, the situation is going to be inverted, because the pressure of CO2 in the blood is 40, while the pressure of CO2 in the tissue cells is 45. So CO2 moves from tissue cells to the blood. So this process, the gas exchange in the alveoli, is called external respiration, and the gas exchange in the tissues is called internal respiration. So this again exemplifies what we just talked about. Okay, so oxygen here enters the alveoli from the inhaled air, and then oxygen enters the blood. You see, blood is oxygenated. Now CO2 moves from the blood into the alveoli and is exhaled. In the internal respiration, the situation is opposite. Oxygen goes from the blood into the tissues, while CO2 leaves the tissue cells and gets into the blood. So why the concentration of oxygen in the cells is lower and concentration of CO2 is higher than in the blood? Cells perform cellular respiration. Cellular respiration produces carbon dioxide. Carbon dioxide accumulates in the cells. Not only that, cellular respiration uses up oxygen, so oxygen levels in the tissues become much lower. Now, how oxygen is transported to the tissues? Oxygen is transported by the molecule called hemoglobin. So here we have an example, that's hemoglobin. Okay. Oxygen binds to the molecule of hemoglobin, and it specifically binds to the iron atom of the complex between heme and iron inside of the hemoglobin molecule. So the hemoglobin molecule looks approximately like this. You have 4 individual subunits. Inside of each subunit there is a molecule of heme, and inside of molecule of heme there is an iron. And that's where oxygen binds to. Hemoglobin can also carry carbon dioxide. However, carbon dioxide does not bind to the molecule of heme. It does not bind to the iron. Carbon dioxide binds specifically to the protein part of the hemoglobin molecule. It binds, let's say, here. Now, with that said, we can take a look at the so-called hemoglobin dissociation curve. As you can imagine, if we have a molecule of hemoglobin that consists of 4 subunits, and each subunit can bind 1 molecule of oxygen, the total number of oxygen molecules that can bind to hemoglobin is 4. But not always all 4 oxygen molecules are bound to hemoglobin. Sometimes it can be 3, 2, 1. If 4 oxygen molecules are bound, we say that molecule of hemoglobin is 100% saturated. 3, 75%. 2, 50%. When we average quadrillions of oxygen molecules in the blood, we get the hemoglobin saturation. This is the number that in the clinical settings is reflected as OSAT, oxygen saturation levels. So the curve shown in black here demonstrates the hemoglobin dissociation curve. Basically, it shows you what is the saturation of hemoglobin at the certain pressure of oxygen. So for instance, in your lungs, where the pressure is really high, hemoglobin saturation will be between 90 and 100. And in your tissues, when the concentration of oxygen is lower, the hemoglobin saturation will be somewhere between 70 and 80. So in fact, this is the amount of oxygen that is delivered to the tissues. This is what you got on that's the amount of oxygen. That's how much oxygen you got on hemoglobin in the lungs. And this is the amount of oxygen that leaves the tissue. It's kind of surprising that not all oxygen is unloaded in the tissues, but that's enough. Moreover, that reserve, so-called venous reserve, this reserve is sufficient for a human body to survive for about a couple of minutes if the person stops breathing, if oxygen stops getting into the lungs. Now, on these illustrations, you see on each graph, you see not one curve, but 3. They show you how hemoglobin dissociation, ability of hemoglobin to carry oxygen, how it changes when blood pH or blood temperature changes. It turns out that when blood pH becomes lower, when person becomes acidic, hemoglobin dissociation curve shifts to the right. And at the same oxygen pressure, let's say 40, saturation decreases, which means that less hemoglobin is bound, less oxygen is bound to hemoglobin, meaning that there is more oxygen unloading. So with the pH going down, oxygen unloading increases. Same story you can see for elevated temperature. When temperature goes up, unloading of oxygen from hemoglobin increases as well. Now, how is, sorry for that mishap, it's not, of course, treatment for angina, how is carbon dioxide transported in the blood? So there are several ways. One, it can bind to the protein portion of hemoglobin, as we discussed, called carbaminohemoglobin. Second, carbon dioxide can interact with water, form carbonic acid, which then dissociates into bicarbonate and hydrogen ion. Bicarbonate diffuses into the blood, and hydrogen ion binds to hemoglobin molecule inside of the red blood cell. In fact, the same process can be seen in the plasma, it's just less efficient. The point is that CO2 can be transported from tissues to the lungs, either bound to hemoglobin or in the form of bicarbonate ion. And bicarbonate ion is critically important for maintaining the blood pH. Bicarbonate ion serves as a buffer, allowing to neutralize small changes if there is a local sort of decrease of pH, increase of pH. Bicarbonate ion can ameliorate, can negate those changes in the pH of the blood.